Okay, our next speaker was selected from poster presentations. For our oral presentation is Emma Sprooten from Dr. Sofia Frangu's lab. And she's going to talk with us about trends and patterns in 15 years of fMRI research in psychiatric disorders.
So again, thank you very much for inviting me and for everybody to come back and to listen to this talk. So I'm going to talk about trends and patterns in 15 years of fMRI research in 5 major psychiatric disorders. And this is only one of the meta-analyses that we did. The other ones in our group and the other ones are presented in the posters, so you can visit them as well. So evidently there has been a surge of neuroimaging studies in the last 15 years, and neuroimaging has already had a transformative impact on psychiatry by establishing the public and scientific perception of mental disorders as disorders of the brain. And so after 15 years of research, we have come to a point where it becomes meaningful to look at all of this data in aggregate. And specifically, neuroimaging in psychiatry is facing 2 challenges, or two of the challenges that neuroimaging is facing there can be addressed by looking at the data in aggregate. And one of them is more methodological. It's how are we going to integrate all of this big data in a quantitative and objective way that we can then use to interpret further, taking into account all of the variables that the different studies use in their methodology and their acquisition of the images and the analysis. And the second challenge is more conceptual, and it's more about how are we going to turn all this big data into knowledge, and how we're going to give it meaning in terms of translational research, but also in terms of just general theoretical meaning for neurosciences. And one way of integrating this data that can help in identifying or help in these challenges is by doing meta-analysis. So we did a meta-analysis of fMRI studies in 5 major psychiatric disorders: schizophrenia, bipolar disorder, major depression, anxiety disorder, and OCD. And we started by identifying 547 studies that used task-related fMRI to compare patients with each of the disorders to controls. And in total, these studies identified or reported 5,957 coordinates, where there were where there were significant differences between the patients and the controls in fMRI activation. And then I mapped all of these coordinates to standard atlases in FSL, and from there I calculated a number of unique studies that contributed 1 or more coordinate to each of the atlas regions. And this number of unique studies per region is the dependent variable that I'm going to continue talking about in the rest of this talk. And using this number and the distribution of the numbers of unique studies contributing per region, you can statistically assess which variables that differ across studies seem to influence the pattern of results that have been reported in the literature. So in general, fMRI studies, or in general neuroimaging studies in psychiatry, tend to address each specific psychiatric disorder in isolation. So they tend to compare patients with one specific traditional diagnosis to a set of control participants. And this is a manifestation of the field in general. So people, or psychiatrists, or in general people, do investigate each disorder in isolation. They're experts mostly for one specific disorder that develop specific theories of the underlying neurobiology that underlies each disorder, and hypotheses that are tested within each disorder. So in general, psychiatry research operates within intellectual silos that are defined by the boundaries of the traditional diagnostic categories. So one of the questions that we can ask by looking at all of the fMRI data that has been published, or the results have been published, is how diagnosis specific are the patterns that are defined, that are found to be different between the patients? Are they actually following these diagnostic boundaries that are assumed from the beginning? So here I've plotted the number of, or the percentage of studies for each of the disorders that contribute to each of the subcortical regions. So schizophrenia is plotted in green, bipolar disorder in blue, and depression in purple, anxiety disorders in red and OCD in orange. And there are two major observations that you can see from here. And the first one is the amygdala and the hippocampus seem to have been reported the most. But if you normalize by region volume, which you cannot see on this figure, but if you normalize by region volume, it is really the amygdala and the nucleus accumbens that have significantly more, reported more frequently than would be expected. And the second observation that you can see here is that in general, in a lot of places, the lines seem to run in parallel, so there are not that many diagnostic-specific patterns visible. And when we looked at this statistically, the diagnosis had a significant effect on the number of studies that contributed to the amygdala, as well as to the caudate. And you can see that somewhat on the picture as well. However, one important consideration in methodology that has already been written a lot about in neuroimaging is the level of inference that has been used to analyze the images. So in particular, about half the studies that we included in our meta-analysis used something called region of interest analysis, whereby the investigation is really restricted to one or more regions that are a priori considered to be of importance to the task or the disorder. So the other regions are just not at all considered or reported on. So this can, of course, have an influence on the region counts or the study counts per region that we calculated. If some regions are not at all considered, they're going to be less often counted. So here I've split up the previous figure by level of inference, or by the studies that use different levels of inference. And you can see that for some of the subcortical regions, in particular the amygdala, there is an overrepresentation of region of interest studies that have contributed mainly to the results in amygdala that is not really supported by the whole brain studies that are less biased, where the amygdala is less frequently reported. For the brainstem, for example, in the thalamus, the opposite was true. You would have expected them to come up more often in regions of interest studies, but apparently people aren't that interested in them a priori, as would be expected on the whole brain level. They come up more frequently than expected. And a second observation that we had when I divided up the whole brain and region of interest studies is that on the previous slide I showed a significant effect of diagnosis in the caudate nucleus and the amygdala, and this was not significant when we only looked at whole brain studies, so it seemed to be mainly driven by region of interest studies. Okay, and then we're going to have a quick look at the cortex, and there's a lot of data in this slide, so I'm not going to go into too much detail, but here, for visualization purposes, I've also just shown the only 3 diagnoses to start with, and this is the distribution of the region counts in the cortical regions. And the first observation is that overall, in the cross table of all of the regions by diagnosis, there was no significant effect of diagnosis, so the pattern wasn't significantly different depending on the diagnosis. However, when we did it region-wise, the analysis, there were some nominally significant effects, particularly in the medial prefrontal cortex and the inferior temporal gyrus, where diagnosis seemed to shift the study count somewhat towards one or more diagnoses compared to the others. But again, though, when we only looked at whole brain studies, there were no significant effects of diagnosis of the activation patterns across the cortical regions. So this is consistent with the subcortical findings as well. So perhaps this is a bit more clear to interpret just visually, because if you just look at major depression and anxiety disorders, the 2 remaining pictures that I hadn't shown, you can see very obviously that they are very similar in terms of their activation patterns in comparison to controls. They actually show very similar abnormal activation patterns when you aggregate them across all the studies. And in fact, when I correlated the study counts per region that contributed to each region across the regions between major depression and anxiety disorder, so simple correlation over a dependent variable, the correlation coefficient was 0.91. So it's a very, very highly significant correlation. And perhaps it's not so surprising that these 2 disorders are so similar in terms of their fMRI. They're also quite similar, they have comorbid symptoms clinically, and also genetically, they are very highly genetically correlated. So it seems that this is mirrored in the neuroimaging pattern as well. So the last main question that I'm going to address here is what is the influence of task paradigm on the observed diagnostic activation differences? And this can be split up in two questions. Firstly, does the choice of task paradigm really influence what we can uncover about case control differences within each disorder? So the task can lead to some regions to be activated, and that might be necessary to uncover the differences between the patients and the controls. And the second question is, perhaps we didn't see any diagnosis-specific effects because they only exist or they are different between the different task paradigms that people use. So does it modulate the disease specificity of fMRI? And to do this, we classified all of the studies according to the task they use in the fMRI scanner, the MRI scanner, sorry. And RDoC domains are a framework that is specifically developed by NIMH, specifically for research in psychiatry. And there are 5 of them: cognitive systems, positive valence, negative valence, social processes, and arousal and regulatory systems, which is not addressed here because it's rarely used in imaging. So looking at the subcortical structures here, the proportion of studies for each RDoC domain that contributed to all of the regions are plotted. And considering all studies, again, there were some significant differences in terms of how the RDoC categories were distributed. So task design seems to have a significant effect on the distribution of the pattern results for the amygdala, the hippocampus, the nucleus accumbens, and the putamen. However, again, when we looked at whole brain studies, this effect was attenuated and wasn't significant anymore. But when you look at the picture, it still looks quite similar. So we do not disregard this at all. It still looks like it's supported somewhat by whole brain studies. The second observation, the second question that I want to answer, and you can't really see it on the slide, but that's whether the diagnosis-specific effects appeared more clearly when you subdivide the studies by task design, and this wasn't the case. So I did all of the analysis, and there are no significant effects beyond what could be expected by chance, given the number of tests that have been conducted for all of the regions and across all of the RDoC domains. Okay, so in conclusion, the analysis that we did does not, in this case, address whether or not a region is significantly activated or deactivated in a disorder, but it rather can address, it does in the first place show that brain imaging has repeatedly shown differences in brain activation for every disorder across many regions. And secondly, what the purpose was of this analysis was really to show which factors that differ across studies modify the results that can be obtained in the last 15 years across all of these studies that have been published. And clearly, on a methodological level, region of interest studies have overemphasized certain subcortical regions in a manner that is not supported by unbiased whole brain studies. And so, of course, there are some good reasons for choosing regions of interest, but in general, there should be some caution in interpreting them, because in general, these studies are designed to confirm the pre-existing theories which have, in the first place, on which they base the region of interest selection. So that's one consideration. And the second conclusion, the third conclusion is that although it seems that neuroimaging may not be revealing the neurobiological mechanisms specifically for each disorder, it is revealing something more fundamental about how the brain functions and is organized and how this relates to mental disorders. So it seems to be that the brain is a less modular organ than some of us had thought before, in terms of the modulation of RDoC domains, for example, didn't really do very much to the data. And secondly, fMRI has limited specificity for the traditional diagnoses as they are currently defined. And this is not an issue that is very specific to neuroimaging per se. It's also found in genetic research. So I think that together, genetic research and neuroimaging encourage studies that investigate neuropsychiatry that do not rely completely on traditional case control comparisons. So I want to thank you for listening. I want to thank everybody in our group and Sophia. And I don't know if there's time for questions.
So before the questions, I would like to give you this certificate of recognition.
Thank you very much.
You did a great job. That was beautiful.
There's also a small financial award that will come in your paycheck, apparently.
Oh my God.
It's small, you won't see the difference. It's going to be there.
You can buy cross samples.
Okay. So after the niceties, do we have questions? So let me ask you, what was the set of inclusion of the studies into your data set?
Right. Yeah, this was for, set of inclusion. So they had to be task related and case control comparisons. And I don't know if there was anything else. Adults? Adults?
Yeah, so there were adults, they have had to have had some sort of diagnostic assessment using, they should have reported coordinates in in a sort of standard space, and they should have used clean population, so not we use a mixture of whatever, so there would have been one diagnosed at a time, and there would have been sufficient information for us to be able to look at moderator variables, like some of which.
And in terms of the cognitive task, was there an active cognitive task and an active control or baseline, fixation baseline task?
Always an active control.
Other questions?
Yes. I'm not an expert in fMRI, but so this may come across just as my ignorance on the field. So you're comparing different groups with task-oriented fMRI, and I'm assuming that the task is specific to each disease, or is it similar in all disease?
So that's a great question, which is partly we investigated because so this was a meta-analysis, just to be clear. So the tasks that were used across all of the studies differed, but they tend not to be, people tend to apply similar tasks across the different diagnoses.
Because that's the difficulty. If you are applying a different task, comparing groups with different tasks, then you are in some way biasing the comparison.
Right.
So it's almost like suggesting that perhaps resting state fMRI may be a more fair study of comparing these different groups and see which one will basically just come across.
Yeah, we looked at the effect of the different tasks according to the Outlook domains, which is the most objective way we could think of classifying them, and that didn't make a difference in the diagnostic effects. So it's something that we tried to look at as best as we could given the meta data that we had.
Thank you.
Yeah, actually, good segue to the other audience. The question whether you look at the resting state, bold fMRI, for psychiatry, that's number one. Number two is whether you look at the DTI, especially kind of the foundation of the brain structure, especially the network. Really kind of the question is how specific such meta-analysis, especially we see some very promising data using DTI to look at deep brain structure in depression. However, I didn't see the structure pumping out from this meta-analysis. So there's something that still missing. Probably we're all aware that the fMRI is based on task related. So we might only look at peak of the iceberg. So something more fundamental maybe need to be investigated.
There's a lot of different aspects to the question. So, well, I would love to look at DTI. Most of my research is in DTI. So, but I don't think there's enough studies published yet to do a similar meta-analysis in DTI. And the other thing is that when you look at maybe specific populations with depression, they might be more homogeneous populations. So you might find that some things are more, reliably found in them. Perhaps is that something? Is your population just general depression, or is that something that could be?
No, what I infer is really try to suggest is looking at the more so-called default mode network, so that kind of thing, because we all know that actually the brain consumes more energy, just maintain the status. That's more relevant to psychiatry, a lot of psychiatry disorders. Rather than evoked as a caveat for that is how you compare apple-to-apple comparison. Is the task different, or how a subject engage in that task is very challenging to standardize. However, the resting state and some DTI may hold some promising.
Yes, I think in the future we should do similar things for resting state studies as well, I suppose. Yeah.
Any other questions? Well, I'll conclude this session then and this talk by just one comment. And addiction and substance abuse would be nice to add to the psychiatry illnesses list. But thank you very much for a great talk, and this concludes our neuroimaging session.